So yes, so as Eugenie said, we're coming towards the end of an Australian Research Council Centre of Excellence programme in plant cell walls. UQ is one of 3 universities involved in that, and we have 2 main areas we've been looking at. Now, normally when I talk to this group, I talk about the nutrition and health aspects. This time I'm going to talk about the biomaterials aspects of primary plant cell walls. So first thing, what's a primary plant cell wall? Well, if you look inside things that we should be eating every day, then you can see a structure which is held together by relatively thin walls around a large amount of liquid, and that is really, those are the plant cell walls. So these features here are typically of the order of a micron thick, and they're typically enclosing cells which are more than 100 microns across. So they're encapsulating a lot of water. There is skin around the cell, but they also have to connect with the rest of the tissue to provide the various functions. One of the features that is a characteristic of all plants is the heterogeneity. So you can easily see that here. We're looking here from the inside, near the middle of the fleshy part of the tomato, out into the skin, and clearly you can see there's a gradation of cell structures and a heterogeneous environment. So if we want to understand the material properties of this, we're going to be challenged because of this heterogeneity. Probably the most homogeneous type of sample you get is like this. So this is a storage organ, potato tuber, and it's still not that homogeneous. So most man-made materials are way more homogeneous than this. So again, that leads us to challenges of how do we integrate from structures and functions at an individual wall and cell level up to features of a tissue. So that's, and this being a relatively homogeneous tissue, is a storage organ in potatoes. Those are 2 things that we eat directly, but also from a point of view of food-relevant cell walls, there are also those structures that we try and extract things out of. So the tea leaf that goes in your tea bag or in your brew has a cellular structure, and the time that it takes for the brew to develop is in large part due to the passage and the transport of the various molecules out of this multicellular structure. So it's not just about physical structure, it's also a barrier to movement, and it's a feature that we can control in order to generate a wide range of different properties, in our case, where we're interested in foods. So these are mostly primary cell walls, and primary cell walls are characterized by a lack of lignin, so that they are characteristic of growing plants and of fleshy tissues of plants. And so from a food point of view, those are mostly what we eat. We don't eat too many of the woody tissues, and therefore our focus in the food and nutrition space is on primary plant cell walls. So we address this from the multi-scale point of view. So there are 3 levels of structure that we have to take account. One is the architecture of the material. That's the material science of the total tissue. The second is the properties of the individual units, the cell, and these are essentially hydrated particles or very large colloids. And then you talk about the polymers which make up the individual cell walls and how they're arranged and how they contribute to the scale of structure at the cell and the tissue level. So we talked already about the heterogeneity at the tissue level through examples of the micrographs. So let's now look at work that's been done here at UQ in order to look at the heterogeneity of the material properties at the cellular level. And in this work, individual plant cells, in this case from tissue culture, are rested in a well, a microwell array. So the cell is sort of sitting in this little depression here, and this allows you to go in with an AFM tip and to measure the mechanical properties of that wall. This is looking down the top of the AFM tip on top of the cell. And this is by no means trivial, and our colleagues in chemical engineering have been developing the analysis regimes, the multi-regime analysis that you need to interpret these force deformation curves. But at the moment, I'm not going to talk about the numbers we get. I just want to show you the heterogeneities that exist. So here on the left is a map of the Young's modulus. That's the compressive strength of the material as a function of, this is nanometer scale, so this is a 5 micron by 5 micron piece, in this case of the suspension cultured cells of a ryegrass. And you can see there's a significant patterning in here and a large, more than an order of magnitude in Young's modulus variation at roughly the micron sort of length scale. So the patterning is about the micron length scale. Now, at the moment, there is no technology where you can directly get chemical composition at the same time as AFM. We're working on it. But if you do things in parallel and look at the chemical heterogeneity of the same sort of material, not the same cell, same sort of material, then you see these sorts of patterning. So this is after we treat with a reagent to remove most of the polymers from the cell wall and leave the cellulose behind. And in this case, we see there's a patterning of cellulose deposition also at the micron sort of length scale. So we think we need to take account of that patterning in order to understand how the whole cell behaves. And it throws us into then a quandary. How do you interpret the mechanical properties of a whole cell if you've got these heterogeneous local zones? So we have to go down another scale again in order to try and understand that. This is not limited to suspension culture cells. Here's a bit of everyone's favorite model plants. This is a Arabidopsis leaf. And again, you can see in terms of Young's modulus patterning, there's about an order of magnitude variation in Young's modulus on a mature leaf. And again, there's no obvious structural features underpinning this, we believe, no obvious ultrastructural features, microscopic features. So this looks like it's also down to variations in the local concentration of the cell wall polymers. So our approach has been to try and understand how we can appreciate those properties by going down to the molecular level and back up to the mechanical level. So if you look at a tissue and say, well, actually a cell wall is actually 2 cell walls, so the boundary between 2 cells, each cell has its own wall, and therefore what you would call a single cell wall between 2 cells is actually 2 cell walls. Not always, but often with a middle lamella, which is usually deficient in cellulose if it exists. So for those cells which are relatively easy to pull apart, they will have a middle lamella because that allows, and that is a zone of weakness for them. Within this structure of an individual cell wall, the classic diagram is like this. And here we have the 3 main components. We have a cellulose rods, thick, and I'll show you, well, and you'll see in a minute why. And then 2 other types of polymers, generically known as pectin and hemicellulose. In this case, it's a particular hemicellulose, xyloglucan. And the model shows that we expect that the pectin can form its own network, so pectin gels, just like it does in jam. And we think that's what happens within a cell wall, so there's an independent network of pectin. And that the hemicelluloses are often associated with the surface of cellulose because they have very similar chemistries and can absorb quite easily to that surface. So we're down to understanding the cell wall as a cellulose-based network. On this length scale, remember we're saying that the heterogeneities at the cellular level were on the 1 micron or 1,000 nanometer length scale. So we're now bridging those length scale divides and able to get to hopefully a coherent description. And then underneath that, so if you look at the individual cellulose rods here, they are crystalline, semi-crystalline, because they're associated with individual polymer chains coming together in a crystalline organization in a way that I'll talk about in a minute. So it's a very complex structure. Remember, it's a very thin cell wall that's encapsulating a lot of liquid, so it has to have a lot of strength. And to try and get these points across, one of the activities we've done in the last year is engage with a video animator to try and develop an educational model that helps us understand this, which I'd now like to show you. Let's challenge the technology. There you go.
The primary wall of plant cells has a composite structure that provides support and protection while remaining flexible enough for growth and expansion. The primary wall is made of carbohydrate polymers laid down in layers and branching networks. This illustration presents a model of carbohydrate structure that includes hemicellulose, pectins, and strong cellulose fibres. The primary wall is created at the cell's outer membrane. A rosette of cellulose synthesis enzymes moves across the membrane surface, generating long chains of cellulose molecules which combine into stiff microfibrils. A vesicle from the cell's interior releases its cargo of hemicellulose and pectin, which interact with the cellulose microfibrils.
Okay, so the picture here is, so we have cellulose is made at the cell membrane. So if we go back here, then we're talking about a structure where this is a cell membrane here that defines the boundary of the cell. The cellulose synthase sits in that cell membrane because cellulose is insoluble. You cannot make it in one place and then transport it somewhere else. It has to be made in situ. So the cellulose synthases sit within the cell membrane. The other polymers that go to make up this composite structure are made inside the cell in the Golgi apparatus and then exported in vesicles, and then they are deposited into the extracellular space around and sometimes synchronous, sometimes after, before the deposition of cellulose. And so it becomes around, the way the cell wall is set up is that as the cellulose is deposited, these other polymers are in the way and interactions can occur and a composite structure can result. So I think that's an important thing to really get to understand how we can mimic that, because we cannot make cellulose alone in the test tube. We have to have a biological system that makes it, but we can choose the environment in which we allow cellulose to be made such that we can study how individual polymers work with the cellulose synthesis system. Do you want to see the video again, or shall we go? Yeah, let's see the video again, I think, because it's quite a lot to take in one go, but it's got hopefully captured all of the elements of it. Here we go
The
again.
primary wall of plant cells has a composite structure that provides support and protection while remaining flexible enough for growth and expansion. The primary wall is made of carbohydrate polymers laid down in layers and branching networks. This illustration presents a model of carbohydrate structure that includes hemicellulose, pectins, and strong cellulose fibres. The primary wall is created at the cell's outer membrane. A rosette of cellulose synthesis enzymes moves across the membrane surface, generating long chains of cellulose molecules which combine into stiff microfibrils. A vesicle from the cell's interior releases its cargo of hemicellulose and pectin, which interact with the cellulose microfibrils.
Okay, so this is really the work of Drew Berry, who's at the Walter and Eliza Hall Institute in Melbourne, so connected through our colleagues in the Centre of Excellence in the University of Melbourne, so Tony and Monica from the University of Melbourne, and it was Drew's voice you heard. So if you go YouTube him, he's got some fantastic videos around medical and health-related issues. This is one of his first adventures in the plant world. So, okay, so now we're going to go back and start assembling that structure and understand the factors involved in that. So, cellulose microfibrils, this rosette. So this rosette is really jet propelled. So the energy of extruding a polymer causes this rosette of enzymes to move the other way. So it's like a jet engine. All right, that's why it moves through the plasma membrane. It's because it's actively it's turning chemical energy into mechanical energy and moving through the cell wall as it produces the cellulose out the other side. But until recently, it wasn't really clear how many chains it made. So we contributed to that discussion through understanding how to interpret NMR spectra and doing molecular dynamic simulations, particularly with Daniel Omer, who was based at the IBM research facility in Melbourne, and showed that it really had to be 18. And just 6 months ago, this was much more rigorously proved by the biologists. So it's pretty clear now there's 18 chains of cellulose in an individual microfibril. But these individual microfibrils can come together. So that was the other work that Daniel published. So the 18 chains in plant fizz, and then in cellulose we published how these things interact and doing molecular dynamics on bringing them together in various orientations. You can see there's just two various orientations. And interesting how sometimes you get water traps, sometimes you don't. And that depends on the orientations that you allow. But there are very different energies involved. So this is around the energies involved in bringing these things together. And you can see there's a factor of 4 difference in energies. So if there's enough time for them to sample different spaces, then they will come to a rather clear decision as to what arrangements to make. But the cell wall, the cellulose synthesis occurs pretty quickly. It's that cellulose synthesis rosette moves at 300 nanometers per minute. So it's pretty fast. And so we think that it's probably more a kinetically trapped structure where there isn't the chance for the thermodynamics to come into play. But this remains to be examined experimentally because we don't have the tools yet to do that. What we do have are our ability to look particularly at the scattering using X-rays and neutrons, which we do with our collaborators at ANSTO near Sydney. And that's particularly suitable for this length scale of around the few nanometers length scale, which is characteristic of cellulose microfibrils. So with NMR and infrared, you can look at the primary structure, but we want one length scale higher, and so scattering is really the ideal tool and technique to do that. And we've done a lot of work with Marta, a postdoc who was based here and paid through here, but based at ANSTO. So this is what we've been doing. So we're developing measurement tools for measuring the microfibril structure. I talked about that, and I'll show you the results for the small angle X-ray and neutron scattering. We look at how individual polymers interact with cellulose surfaces by doing surface force and adhesion measurements. I'll talk about those. We're also interested in what is actually the major component of primary plant cell walls, which is water. So these are highly hydrated environments, so water is the single largest component in a primary plant cell wall, often ignored, but has a really important role to play in terms of fluid flow and what is known as poroelasticity, which I'll explain to you. And I think I've already gone through the local heterogeneity question. We've shown that in the biological system. How do we get around that in our model systems? So if we really want to get to the structural basis for materials properties, we have characterized the interaction between cellulose for all major types of plant cell wall polymers, developed a poroelastic model which integrates the role of water with the role of polymers in determining structural features, and we've linked the properties of defined cellulose composite properties to plant cell walls. So that's what I'm going to talk to you about. Because we're moving towards this 3D multi-scale model, so that animation you saw was part of that, so we can try and take that a bit further and start to understand how it behaves at the tissue level. So this is a normal sort of frighteningly complex structure that will be drawn by biologists and biochemists because we can measure all these molecules. There are, if you really wanted to look at differences, there's probably around 40 or 50 different subclasses of polymers within the cell wall. But there's really only 3 that you really need to know about. So the pectin family, the hemicellulose family, and cellulose. So this is normally what people would say, well, okay, look at that complex structure. Surely every polymer has a specific role. This is nature. Sort of biology has designed each polymer to have a specific role. We don't think so. We think a lot of the polymers from a material point of view are redundant or are leftovers from an evolutionary process which no longer needs them. So our hypothesis is that although the composition is complex and heterogeneous, the guiding principle is about managing the properties of cellulose, because cellulose has the very effective strength, but is also impossible to turn over. So other polymers can be turned over with enzymes. Cellulose, basically, once it's laid down, is there and is not really turned over. So therefore, cellulose is what you have to manage, but cellulose is what gives you the really efficient strength, per unit energy or volume. So that's our thesis, and that's really what we've been working through. So the way we do this is we say, well, fortunately, it's not only plants that have these rosettes that make cellulose. There are certain bacteria that also do this. So they have just the same synthesis machinery in their outer membranes that produces cellulose in microfibril arrangements, but in a fermentation system. So we have control. That's really important, and this is those of you who have either traveled particularly in the Philippines. Nata de coco is a very popular jelly dish. Those of you, I think you can still get them. You used to get a lychee drink here which had pieces of jelly in it. That was nata de coco. That was cellulose. That was cellulose made from bacteria. So it's a traditional food fermentation process, and it produces hopefully you can see sort of pieces of cellulose gel that you can pick up with tweezers, multi-centimeter scale, and that suddenly allows us now to do mechanical tests that we can't possibly do at the micron level that we would need to capture the heterogeneity of the plant materials. So the bacteria have this same sort of rosette structure, which down here, which produces the cellulose chains, which come together to form fibers and so on. But this is a fermentation system, so we can choose what we put into the fermentation medium, so we can study what happens when cellulose is produced in the presence of, let's say, pectin as a solution, pectin as a gel, xyloglucan, whatever. We can choose what we put in there. So we can mimic the assembly process of plants in a bacterial fermentation system and produce materials that we can then study at the mechanical level. And so, of course, we've done that. The sort of structures you get, this is in electron microscopy. With cellulose alone, you get these bundles of fibers come together. No obvious orientations, but some bundling that goes on. When you grow it, the cellulose in the presence of certain other cell wall polymers, in this case arabinoxylan, one of the main serial cell wall polymers, you see these deposits on the surface of the cellulose, and almost certainly, we know that those are arabinoxylan because you can see them in a transmission electron micrograph. You can label it up with immunolabels. And for another example, xyloglucan, another type of hemicellulose. In this case, you can see, I've pointed them out with arrows, hopefully you can see them. You get thin-stranded cross-links that bridge across the cellulose fibers, and this is what had been seen in plants as well before. You can probably see some of them at the back here, thin strands that are cross-linking the cellulose fibers. So these are structures which are spontaneously assembled because we're just secreting cellulose into a medium that contains, in this case, solutions of arabinoxylan or xyloglucan, and this is the physical chemistry that occurs and the structures which are generated. And these are the structures that we've looked at in detail with small angle X-ray and neutron scattering. I won't go through all of the information we can get, but the key factor here was when you look at the arabinoxylan composite, the one which had these nodules on the surface, we can show that the arabinoxylan molecules are only accessing the very outer edges of the cellulose fiber, whereas the xyloglucan with the cross-linking, we can show that the xyloglucan is accessing the interior of the fiber, and that this means that they must have interacted, the xyloglucan cellulose interaction must have happened before the cellulose fiber was finally formed, so at the point of synthesis. So this is why the timing becomes critical. If you have xyloglucan there at the point of synthesis, it gets incorporated into the fibers and can then cross-link them. If you add xyloglucan afterwards, it can just sit on the outside. It doesn't have this ability to get inside. And so we've found new ways of measuring this, which is great, and got a lot of papers out of the small angle X-ray and neutron scattering work. So those are two examples of the type of structures that we can make. So we can get molecular cross-linking with xyloglucan. So here the thick ones are cellulose, the thin ones are the non-cellulosic polymers. Or we can have surface absorption, which is stable to washing. So these are thoroughly washed before we analyze them. So it's not just completely weak, not just weak, non-specific association. There's something going on there that's stable to washing. But these are not the only type of structures you can and indeed do get. There's a whole range you can think about getting. So as well as cross-linking, you could have two separate networks, or you could have two networks that are separated, and those will give you different mechanical properties. And you can have surface absorbed materials which are stable to washing. You have those which are surface absorbed but are removed by washing. And we find examples of all 5 of these when we look across the family of the different cell wall polymers. So we say that the way that nature uses the polymers to make cell walls for different purposes draws on this collection of interactions which is available to it. So to give you examples, just put the names up there just so there's more than one in each type. So it's not just xyloglucans, there are certain galactomannans and the arabnoxylans, other hemicelluloses, but we've also shown can cross-link cellulose in terms of interpenetrating networks. Mostly that's with the pectin molecules because pectin can form a pre-gelled system. So if you deposit cellulose into a gel, a mesh network, then the cellulose is kept apart from itself because it has to go through the pores in a network, so it doesn't have as much opportunity to cross over itself, so it keeps more local alignment. Remember that one, that becomes important later. But also, it's not just pectin. Some of the hemicelluloses can also form their own network, and we can deposit cellulose into those and get this same effect. Phase-separated networks occur, so generally that's where the strength of the non-cellulosic network is high. So if we have strong pectin gels, then the cellulose cannot deposit into it, and essentially the cellulose pushes the gel away from it. And the same thing can happen with some xyloglucan, etc., with other molecules where you have promoted the gelation process. And if you remember back to middle lamellae, so this is a model for the middle lamella. So if the middle lamella, if there are polymers in there which are too strong for the cellulose to deposit into, they're going to be pushed to the middle lamella. So this is possibly how middle lamellae arise and are cellulose deficient. Surface absorption, so it's not just in the serial polymers, not just the arabnoxylans, but the mixed linkage beta-glucans do the same thing. But there's also a whole range of other molecules which absorb onto the surface, but which are relatively weakly bound because you can remove them by extensive washing. And this includes the side chains of pectin. It includes pectin under non-gelling conditions, so without calcium. So without the gelling conditions, the pectin still absorbs on the surface, but you can wash it away. And you can with the various arabnogalactan proteins, gum arabic, and so on. So essentially all classes of primary cell wall polysaccharides interact with cellulose. And the point that has been missed by many people is that this latter one is actually probably relevant because we may remove by washing, because that's what we do in a lab. You'd want to remove non-specifically bound materials. But the cell wall ain't washed. There's no washing process in a cell wall. So we would say that this would suggest that essentially the surface of cellulose is essentially always covered in a cell wall. And that's what we tried to capture that in the video as well. So it's not a question of occasional cross-links. It's a general coating is the normal thing. So, okay, so what does that mean in terms of material properties? Okay, so this is where we get into the word of the day, poroelasticity. You'll be able to amaze your friends with this afterwards. So this is where you have a fluid phase and a solid phase, and the fluid phase components play a role in the mechanics. So imagine you have a sponge filled with water. If you squeeze that sponge slowly, the water will just all come out. But if you hit that sponge hard, it will be hard. You have not given the water enough time to move out of the way, and water cannot be compressed. Water is incompressible. So depending on the rate at which you interrogate something, hit something, then you will have a solid response or you'll have a very soft response. That's at the essence of poroelasticity. I used to give the example, but we don't get 10-meter diving boards anymore in swimming pools. They're too dangerous. A great example of poroelasticity. You go off a 10-meter board, the strain rate at when you hit that water, it's solid. Right? That water is not getting out of the way quickly enough for you. But if you just gently get yourself into the pool, there's no solid character at all. So that's slow versus fast. So this is an important concept, we think, here. And what can affect that is not just the strength of the polymer network, but the viscosity of the liquid phase. So if you've got a viscous phase, that's not going to move out of the way so quickly either. So a viscous solution phase or a strong solid phase, the interactions between those really determine this poroelastic response. So how did we measure that? Well, this is in Jason Stokes' lab, a very neat way of doing it. We just do sequential compression and then measuring the strength. So we compress at different rates, stop, measure the strength, press again and stop and measure again. What is shown here are what happens when you do it at 3 different rates. So you get higher responses with the faster rates for this reason. And these rates, we're now going 33 microns a second, so it's not exactly fast. Right? Microns per second, that's really, you can't see that. So this is really not fast. And you're getting significant effects there. You're showing the effects of the fluid phase on the overall material properties. And so we've done a lot of work in this. Mauricio has got a model for this, and we published the initial paper in Biomacromolecules on cellulose alone, and then this second paper in PLOS One was all the effects of other polymers as they interfere with this poroelastic behavior. So we think about what plant cell walls have to respond to. So compression is one of the things they have, because they have these balloons of water that are pressing against each other, this so-called turgor pressure. And that's really what they need to respond. So compression is really what they're about. And what you've got here, though, is think also about compression in terms of response to environment. So if an insect lands on a leaf, that's actually a pretty strong force to a leaf, and that will happen quite quickly, and that will appear strong. If you put it on slowly, it will appear quite weak. And so it's also about responding to the environment here. So that's the poroelastic model. Another thing you really need to understand is how things move past each other. So this is interfacial behavior. So this is the science of tribology. And again, with Jason, we have a world expert on site here, so it's been great to work in this space using his knowledge in this. So the experiment here was we took 2 of these gels, of these hydrogels that we got from the bacterial cellulose, and just put them together and forced them to come into contact. And then we twisted one past the other. So we're looking at the sliding or the friction between 2 sets of hydrogels. So there's 2 important things. One is that you measure the force that you need to cause those things to start sliding. So that's the interfacial yield stress. And then you measure what happens as it does start sliding. So the interfacial yield stress behaved very nicely. So we're using our 3 model systems of cellulose alone, cellulose with rhabnoxide, and cellulose with xyloglucan, so the surface absorbed and the cross-linked. And if you measure the modulus, that's just the intrinsic modulus of the material, the elastic modulus G prime, that scales with the interfacial yield stress. So that shows that it's a well-defined property of the strength of the material. But what is different, and I hope you can see it up here, is that we've got 2 different types of behavior. So this one, cellulose xyloglucan, the dashed line, when it starts to slide, it just slides very smooth. You get a very smooth response. With the other 2, when it starts to slide, you get this jagged response. And this is called stick-slip. So it slips and it sticks. And then you have to push again and it slips and it sticks. Whereas the xyloglucan, as soon as it starts moving, it's a nice, smooth, gentle. So it's a lubricated event. Whereas the cellulose alone, cellulose rhabnoxide, is not a lubricated event. It's a stick-and-slip event. So that was an important concept, and I think that's really, we'll come back to that one, but that's one to really remember, because this is about how the polymers move with respect to each other, and this is about plant growth, right? So when plants grow, the cells have to extend because you don't make new cells, you generally grow the current cells. So the cell wall has to extend, and so you need to have these ability to slide seamlessly. If you had stick-slip, that would be disastrous for a plant, and I'll show you later what happens. So another way you can look at this is go back to the AFM and start looking at a way of characterizing individual polymer-polymer interactions. So this was the PhD work of Grace Dolan, particularly with Gleb Yakubov, who really developed this technique, the so-called dip and drag technique. So here's the surface of one of our cellulose hydrogels. We've now dipped we can see there's a loop sitting out here enticingly. So we put the AFM cantilever needle into this gap and just pull at it. So it's pulling it out of the network. And what you're measuring then is, as you pull that out, you're measuring the breakage of cellulose-cellulose interactions. This is pure cellulose here. So what you see is a traces like this. So as you pull it, it comes to a point where there's okay, I'm now pulling against the cellulose contact. You then have an increase in the energy needed, and then that contact is overcome, and we move on to the next one. So we can measure things like obviously the force involved here, and the distance, so the so-called peeling distance. So how long does it take for that contact to be pulled apart, or does it go very quickly? So that's, we say here, so the energy within that fiber-fiber contact, you have to think about the adhesive energy, but you also have to think about the deformation energy, so what's holding it there and what allows it to move. So arabinoxylan increases the adhesion between fibers, and remember this was the one that caused stick and slip, so this is about increasing the stickiness and therefore needing to overcome that with a slip. Whereas, interestingly, the arabinoxylan also caused a more rapid propagation. So when it did slip, it went more rapidly, but it stuck and then it had to slip rapidly. But the xyloglucan didn't cause an increase in adhesion, but did cause a more rapid propagation, so it did slip easier. So again, now we have evidence at the really molecular level that this sort of stick-slip versus smooth slide is really happening for individual polymers in a different way. So this sort of lines up with work we've done a long time ago in using these systems to look at the mechanical properties. We're now moving into the third and final bit of what you need for a plant. You need to be able to extend. If you think of it in terms of a shoot growing, that's essentially a one-dimensional extension. So the very simple way of looking at this is just to take a piece of material and stretch it, uniaxial extension, and you measure a stress and a strain. So you pull at cellulose, you pull, it's quite stiff. So less than 20% strain, so that's less than 20% extension, the thing will break. But what we see with our xyloglucan cross-linking composite is that you get this very weak structure which extends to much higher. This is 50% extension. Sorry, I'm missing some things. Okay. So decreased stiffness, but increased extensibility. When we treated this cellulose xyloglucan, this is one with the cross-links, remember, we treat it with enzyme which can break those cross-links. It doesn't touch the xyloglucan that sits on the side of the cellulose, but it does break the cross-links. Then you certainly had an effect on the stiffness, but you still retain this extensibility. So the cross-linking was part of the story, but only part of the story. There's more to it than that. And then when we go to the case of cellulose, pectin, so this is the case where we make a gel of pectin, put the cellulose into it, so we expect that the cellulose will be more aligned and therefore potentially easier to pull. And we find that, so the stronger the gel, so low DM pectin gives strong gel, high DM pectin gives weak gel. The stronger the gel, then the more aligned the cellulose is, because the less options there are for those cellulose to come into contact with a strong gel. And so what you have here then is a similar sort of thing with a high extensibility and low stiffness. But the experiment you can do here is you can, because it's only cross-linked with calcium, you can add a chelating agent, CDTA or EDTA, remove the calcium. The pectin can be washed out then, and you can measure the mechanics again, and the mechanics do not change. So this mechanical test does not change when you remove the pectin. So it's 100% down to the cellulose and the way the cellulose is oriented. Okay, so I think I've over sort of cooked that argument, but I think the cellulose orientation, really important. Cellulose contact's really important. So this is where we summarize it and say, how do we put that together? So I guess it's an obvious one. The cellulose density is obviously important. The amount of cellulose you have per unit volume, I haven't emphasized that, but that's clearly important, and we can measure that, and it scales in the way you'd expect it to. But the anisotropy is also important. So the anisotropy meaning how aligned are the cellulose fibers. And so the more aligned, so in this example, we've actually said that, e.g. in the presence of pectin, you'll get a more aligned structure. And that reduces the number of contacts between the fibers per unit volume. And obviously, if you have more fibers per unit volume, you increase. But you'll have, if you have the same number of fibers per unit volume as this one and this one are, if you have a random orientation, you get many more contacts between the fiber than if you have a more aligned structure. So these contact points. So contact points are pretty critical in terms of determining the mechanical properties and the ability of these systems to slide and slip. But it's not just that. We also have to take account of the nature of those contacts. So we call these hard and soft contacts now. So we have so-called hard contacts. We classify those as being cellulose alone, as an example, or as the material is being put under tension or compression, where it's the other polymers might be pushed out of the way. At the contacts, if the contact surface is cellulose, we classify that as a hard contact. If the contact surface is a cellulose to typically a hemicellulose, we call that a soft contact, because the hemicellulose will also have a lot of water associated with it, so there'll be a lubricating contact. So it's an adhesive or a lubricant, and that's what causes, that's the characteristic of that contact. And the consequence for that is that when you try and stretch these things, you end up with needing to break crosslinks in order to extend structures any further than this. But here we anticipate, or I think we've shown fairly directly, that you can get this sliding approach if you have the right molecules present within, between the fibres. And for those of you who are in the sort of plants area, then expansins are nature's catalysis of plant growth. It's looking quite likely that that expansins catalyze this process somehow in a way that's still not quite clear. And I think now this is nowhere near as good a movie as the first one, but it's just to illustrate, and there's no sound on this one. But at the bottom here, what we've got are two cellulose fibres trying to move past each other. We call that a hard contact. But if you have other polymers that are trapped between them, you can see that the mobility of these polymers just makes it easier for that sliding to occur. So these models are not just, these are based on real numbers in terms of flexibilities of molecules and surface interactions, as far as we can tell. So we think that's another way to illustrate that behavior. Actually, let's show that one again, shall we? I'll just go back and do that. Yep. So these polymers don't have to be rigidly stuck to the surface of cellulose, but they have to be there when the tension is at that junction between the celluloses. And if they are, they lubricate them and they allow them to slip, and plants can grow. Oh, it's a bit more than that, obviously. But so, as an example on this, so people have been looking at this from the point of view of what are the molecules required for cell extension, and all of the plant biology is done in Arabidopsis because of the mutant populations which are available. So this is what happens with a mutant where xyloglucan is analytically absent. So the genes that synthesize xyloglucan are mutated, they're not present, so there's no xyloglucan. So there are only hard contacts, we would say, that are available to this plant to grow, whereas in the wild type you've got the soft contacts, the xyloglucan is still there. So hopefully you've already picked up that here you've got a lot more alignment of the cellulose, and here you've got much more crossover of the cellulose. And so we say that it doesn't matter you've got that here because there is xyloglucan there to lubricate. But here, in order to make any sort of plant that might grow, you've got to keep the cellulose aligned, otherwise the thing will stick and it won't be able to slide and grow. But even with that sort of architectural adaptation, so this is an architectural adaptation to a genetic mutation, even so the plant's pretty sickly, but at least it does grow. So it's not enough, so these hard contacts are still going to be limiting that growth, but nonetheless the plant has adapted sufficiently that it can at least produce something, even if it's not standing up so well. So that's it. So it's a bit of a summary now. So first point was that we really need to think around the plant as having zones of the order of microns in terms of cell wall structures, and that makes it really difficult to understand how a whole tissue will behave because of that local diversity in structure that's present. We are still confident, well, I think it's pretty obvious that cellulose has dominant mechanical properties, but I think that the roles of non-cellulosic polymers in shaping the way the cellulose behaves, we see nothing that goes against that hypothesis, and that it's about managing the cellulose, and that you can manage that through aspects of individual fiber width and length, and we're still struggling to measure the lengths, but we'll get there. Fiber density, clearly that's a concentration effect, and anisotropy, which is the orientation effect, and other polymers have a really big effect there. And the nature of those cellulose surfaces, are we talking about hard or soft contacts? So we think that the non-cellulosic polymers, they directly influence this anisotropy, depending on, so whether the cellulose is kept apart or brought together, depending on the different polymers which are present. And the surface chemistry of those polymers determines whether they can absorb and remain on the surface of cellulose and be at the point of local tension when mechanically stressed. But don't forget the role of the liquid phase. So the aqueous phase rheology becomes really important as well, because that is the mechanical role of the liquid phase that combines with the solid phase to give you the overall material properties. And I guess the point from a biochemical and biological point of view is there are many different ways in which you could put together different combinations of non-cellulosic polymers to get pretty similar end results in terms of material properties. So we come back and say, well, actually, the cell wall components are relatively redundant, which is good news. There's plenty to select from when the plant is under stress or under different environments. And you don't have to have a unique polymer to get a unique structural outcome. So just remains me to thank a whole team of people, I hope I haven't forgotten anyone, who are involved in this under the Cell Wall Centre of Excellence, which is involves the University of Adelaide and Melbourne as well. So we've worked closely on this with our University of Melbourne colleagues, and Daniel, who was based at IBM, and Drew from the master animator. The scattering work was done with Elliot at ANSTO, and the rest are a combination of QAAFI people and chemical engineering people. I think a nice combination of experimental and theoretical work that hopefully has taken us a bit further in understanding how do we relate the material properties of plants back to their molecular composition. That I'll finish and welcome your questions. Thank you.